So first, I just want to thank the review panel and the leadership for supporting this project. I have to see that, I was, really impressed by the review panel. It is very clear. They're all like expert in the field. Their comments are very insightful. Really appreciate all the hard work that, they put in here. So I'm, really excited to share what we propose to do, but also hope to inspire some new ideas, new collaborations here. It was awesome to hear from the fellow awardees as well for all this exciting project. Learning a lot this morning. So I'm here to speak today on behalf of interdisciplinary team across School of Pharmaceutical Sciences, School of Pharmacy, Pharmaceutical Sciences, and also School of Medicine, that I think we have the expertise and resources to enable us to get this going very quickly. So Dr. Farsal will provide various reagent and also the pseudovirus assay for our single cell assay development. And through Dr. Farsal, we also have access to biosafety level 3 labs, when we get to that point. Dr. Filner, of course, is the director of UCI's vaccine R&D center, and he's going to provide us a key reagent in terms of B-cell enrichment, as well as, insight for vaccine development. So we have also engaged a company, Emberstone Biosciences, a company I co-founded. So they're going to provide extensive expertise in therapeutic antibody discovery, development. So they're going to really help us to advance this technology to serve a broader community in a very short period of time. So just some more disclosure here. I mentioned Emberstone already, but there's another company I founded as well, Velox Biosystems. You heard earlier from Nick. So Velox is helping Nick and also Filner's lab to really scale up testing, really take their testing to the market. So it's something we're just, really excited about. So, I think in less than just 20 years, we have already witnessed 3 outbreaks of zoonotic beta coronavirus, right? So SARS-CoV and also MERS, and also, most recently SARS-CoV-2. I think the SARS-CoV-2 really represent another cautionary tale, as this likely to emerge from a recombination between a bat beta coronavirus and a pangolin homelock, right? So, and this lead to a new variant strain that's highly fit for human transmission. So, we know many pharmaceutical companies are racing to get vaccines, but I think there's a well-founded caution in the field that these vaccines and therapies may not work as well as we hope for, because the virus will continue to evolve. I think HIV vaccine is a good example of how long it might take, for vaccine development. And I think even for flu vaccines, every single year, so we have to come up a, kind of a slightly new version for the vaccine, because, this virus continue to evolve. I just feel the threat of COVID-19 might be even bigger than we think, because of this continuous evolution and recombination of this virus. Indeed, I think in the first couple of months already, I think more than 5,000 SARS-CoV-2 mutants have already been isolated. The challenge here, I think the biomedical field is not prepared for such a worst case scenario, where the conventional vaccines and therapies approach, may not, work as well as we hope for. So this is really the motivation, motivated us, in this project to develop a modular and adaptable therapeutic platform that can be quickly deployed to develop therapies and vaccines, so we can really match the rapid evolution and emergence of new virus strains. So the platform we've been working on is a high-throughput single-cell system that can interrogate functions of tens of millions of individual immune cells, specifically B cells in this case, in a single experiment. And this is important because this allows us to identify the so-called super broad neutralizing antibodies, which typically exist at a very low abundance from a large diverse B-cell repertoire in patient or from immunized animals, right? And that cannot be efficiently discovered with conventional screening approaches. So the idea here is, once these antibodies are identified, So they can be used as therapeutics by themselves or as vaccines, in the form of synthetic RNAs or engineered AAVs. So let me walk you through how this system actually works. Okay, so you start with B cells isolated from a recovered COVID patient or immunized mice, and you co-encapsulate them, with reporter cells and acid reagent into this kind of a water and oil droplet at a single cell level using high throughput microfluidics. And now each drop serves as a microreactor to interrogate the function of the cells, which in this case is the ability of the B cell to secrete antibody that can neutralize SARS-CoV-2 infection. You can detect the positive droplet using fluorescence and sort them very much like a fax, except in this case, so they're sorted in droplet. So individual droplet can then be dispensed in 96 wild plate, where you can perform single cell PCR and sequencing to determine the antibody sequences. And that's going to be followed by antibody expression and functional validation. So we're really excited about this single cell screening platform because I think it's really represent a new paradigm in antibody discovery in terms of its high throughput and functional screening at a single cell level. And all of this enable us to interrogate a large immune cell repertoire to identify the rare clones of interest. And this platform has already been established and streamlined, and we've been using this already for cancer therapeutic screening. And this actually really enables us to quickly repurpose this, for COVID-19. So, I think a key innovation of this work is the single cell functional assay format, which our lab is really specialized for. And just give you some flavor here. And this is from a different study where we use T-cell reporters to interrogate interactions between TCR engineered T cells, with tumor cells. In the effort to screening for, therapeutic T cells for cancer. Here, as you can see, so here we engineered a TCR T cell reporter, and when these cells bind to the cognate antigen presented by MHCs, by APC, this is going to activate downstream EGFP. Okay, so we can see a fluorescence, right? So as you can see here, so it is a published example. So when the T cell that bind to the cognate APCs, in this case is the NY-ESO as a proof of concept, and you can see at a single cell level, T cells is active very quickly within just a couple of hours. So we can identify those cells, and then we can further develop into a therapeutic T cell. So for this work, what we propose to do is to convert this sort of viral infectivity assay to this single cell format, so that we can interrogate and identify B cells that can produce antibodies that can block infections to host cells. And because we can do this in a multicolor, multiplex fashion, we can interrogate antibody cross-reactivity to multiple viral variants in a single experiment, so that we can hopefully identify super broad or broad spectrum utilizing antibodies that can work for a larger population. To our knowledge, this is the first example of single cell based functional viral infection assay. So once developed, this innovative and powerful assay can be used for screening antibodies for other and emerging viruses as well. So here are the aims. I'm not going to read for you, but basically, in aim 1, we're going to focus on single cell assay development and validation. And in aim 2, so we're going to evaluate and optimize the single cell platform for screening natural large B cell repertoires from immunized animals and patient samples as well. Really try to discover neutralizing antibodies against SARS-CoV-2. We have already started to procure relevant materials and reagents. The Pharmaceutical lab has some of those, and some of the focus I think on campus, and it has been really helpful as well. So we have also submitted a request to ICTS and COVID-19 biobank for the patient samples. So we're really excited to get this going quickly. Obviously, this will take some time to develop a drug, but we hope we can get this single cell platform ready in the next couple of months, so we can share with the community and empower academic and industry partners to expand and accelerate their therapeutic and vaccine pipelines against COVID-19. So with that, just want to thank the COVID-19 review panel and your support on this project, and I'd be very happy to take on any questions if you have.
Okay, thank you very much. So I have a question. I wasn't sure how you recover clones for producing the neutralizing antibody. Is that outcome just recovering the sequence of the neutralizing antibodies?
Yeah. So we so the key feature of this assay is functional, right? So whenever we see a droplet containing a cell that can produce antibody, that can block this infection. So we can sort that out, and then we can perform single cell sequencing. We're going to identify antibody sequence, right? And then we can express that and produce more, so we can validate them further. Yeah.
So I guess the part I'm not following is how you express more once. I mean, you no longer have that cellular material, right?
Yeah, but you know the sequence now, right? So you have the sequences, yeah.
So you would produce them as a synthetic monoclonal, or?
Right, yes, exactly.
Okay, cool. Do we have other questions? Well, I'll ask another question then. So what's the sensitivity?
Yeah.
Have you, from your cancer work, do you have a good sense of these antibodies, as you point out, could be pretty rare even?
Yeah. So what we have shown so far, we're able to recover, I think at the range of 20 clones out of 1 million background cells, for the positives. Yeah, so it's pretty sensitive, and because we can screening a very large number of cells. We typically screening for millions, tens of millions in a single experiment. And yeah, no, I mean the goal here is really to kind of recover those rare clones. Yeah.
This is Sid. Could I ask a question here?
Yes, Sid. Yes, please.
Weian, thanks for this very interesting study. This question is for you, but also for maybe several of the other presenters, Greg or Phil, others. There's a number of potential point-of-care microassays that are under development here at UCI. I presume that's going on in a bunch of places, that others are seeking these rapid, accurate, sensitive assays. Is there a process for comparing them, for determining which ones are going to be most useful in which situation?
Yeah, Sid, that's an excellent question. And I think the antibody testing right now is what people regard it as the Wild West. There's lots of those products on the market. We heard a lot about those. But the caution here is that many of those are not accurate. And you heard from Nick earlier, because they tend to only test 1 or 2 antibodies, and they do not really have the sort of specificity, sensitivity to be accurate. That's why what make Fieldner's marker rate system so powerful, because you can profile such a large number of antigens and antibodies. But getting back to your question, so we submit a EUA, right, so emergency use authorization to FDA right now. So they become a little bit more stringent in terms of antibody testing. And you will need to compare to the RT-PCR results, right? So for a positive sample, you already know that's positive. For negative, you already know that that's negative. And that's kind of the data that they're looking for. And unfortunately, at this point, because everybody tried to race, to get to the market, and there's very few studies or effort that aim to cross compare between these testing platforms. So that's me happy in the years to come. But at this point, there's very few studies, actually do that. Maybe Craig, can elaborate, Nick as well. But that, that's my opinion.
Yeah, it's early days really for cross comparing. And there was one really good study out of the Bay Area that looked at a whole bunch of different test regimens, I think largely for the RT-PCR. And that was extremely useful. So I'm expecting something like that to emerge eventually for antibody testing as well. It's an easy way to get a really high profile, great paper. So it requires someone to have all those tests available. It's probably good for a couple of universities to team up together to work on it. You have to have all those tests available. You have to have a CLIA laboratory, et cetera.
I think one of the things to bring out is that we collected a lot of classified specimens from pretty much as early as you could get specimens, because we were collaborating with a blood bank. We had a collaborator there, Michael Bush. We've been working with him on projects for several years. And so with the coronavirus outbreak, he was very active. And one of the things they do at that blood bank is collect plasma from convalescent cases that get administered then to people who are being treated for coronavirus. But what that put us in a position to get a lot of well-classified coronavirus specimens. And so for our university to have access to those, that will, I think, accelerate these projects and give them even a better chance of a successful outcome. And that's what I think our lab is involved in a lot of the projects here, but I think one of our main reasons for being involved is to provide, some of these well-classified patient specimens.
Okay, thank you. So that opens, that is really an interesting and helpful observation. So if someone is interested in having access to these classified specimens, then would they join your IRB with a modification, or or is your IRB general enough that
yeah, the specimens that we have from the blood bank have already been consented for future use. So they're not, there's not restrictions on their use for other research projects that we have going on here.
Okay, thank you.